ورحمة الله وبركاته. أنا يوسف هوساوي، اليوم حكون معاكم في هذا الويبينار اللي هو بعنوان Application of Next Generation Sequencing in Precision Medicine. طبعا في هذا الويبينار أنا هقدم overview about the DNA, RNA and protein. Then I'll speak about the PCR and the molecular aspects. Then I'll talk about the next generation sequencing. I will focus a little bit about targeted sequencing. Then I'll speak about sequencing in gene therapy and gene diseases, gene diagnosis. Then I'll speak about single cell sequencing technology, the new era. So this is my disclosure. So please feel free to capture, take photos. And if you have any question, don't hesitate to write your question down. And at the end of my presentation, I will be more than happy to deal with any question you might have. So objectives of my presentation is first to understand the basic of variance identification. So how scientists and clinicians and people working on sequencing, how they identify different variants. Then I'll highlight some concepts about genetic sequencing. Then I want to advance your knowledge in NGS and its applications, sequencing and some objectives. And then I will talk about data analysis and some future perspectives. So let's start with some few words about DNA and RNA. And usually, if you want to understand something, then you have to go to the history and learn how it started. So years ago, and exactly in early 1869, there was a scientist called Frederick. And he was believed that there are some materials hold secrets of the humanity. And therefore, he started trying to collect that materials from lymphocyte. And he found some difficulties to collect that materials from lymphocyte. Then he shifted from lymphocyte to leukocyte cells, start developing different protocols. And he noticed that materials is precipitated when there is an acidic media and dissolved in alkaline. And that is the reason why they call it acidic material. Now, since then, they start thinking about this material seriously. And a student of Frederick came and developed several protocols. And finally, he managed to collect that materials. And because it was precipitated in acidic media, so they call it nucleic acid. And since then, we have what we call it RNA and DNA and protein. And this picture, actually, the real picture for the lab where they made the first discovery of the RNA and DNA materials. Definitely, when I talk about genetics, then definitely I have to mention the father of the genetics, where we should take you back to early 1866, where the Gregor Mendel set up the principles for the genetics: law of segregation, law of independent assortment, and law of dominance. So now Gregor Mendel died, and his students, Thomas Morgan, came and revisited his theory and proved the role of chromosomes in hereditary, and he won the Nobel Prize. Now, since this era, actually a new era started. Sorry, I can. So since this a new era started, where in early 1950s, Watson and Crick, they discovered the structure of the DNA. In 1960s, then the researchers discovered the genetic code. 1970s, the Sanger discovered the first sequencing, and 1980s, then Karimalos discovered the PCR, and since 1990 until 2003, the Human Genome Project was started, a collaboration between several countries to discover the map of the genetics. And since 2003 until 2013, a new era in medicine started. And 2013, then personalized medicine started and precision medicine started. And now we are living in new era in medicine based on the genetic concepts. When we talk about genetics, then definitely we will talk about the cornerstone of the genetics, which is the DNA, deoxyribonucleic acid. And this deoxyribonucleic acid consists of three parts: five-sided sugar, phosphate group, and nitrogen base. And as you all know, we have adenine cytosine, and we have guanine thiamine. And adenine is connected with the thiamine, and cysteine connected with the guanine. Now, definitely, you want to know more about the DNA. And this DNA is a double helix, and this double helix have two double strand, while the RNA is a single strand, and then we have the protein. Since the day they discover the chromosomes and they map the human chromosomes to human and link these chromosomes, different chromosomes, to different diseases, that was a big breakthrough in the world, because now the people, they start understanding the compositions of the chromosomes and link these chromosomes to several diseases according to their position. And several discovery was made it, and sorry. So several discovery has been made, and the outcome of the Human Genome Project give more than 3 billion base pair. And the scientists discovered by that more than 23,000 of protein coding genes. If you look to this slide, you will find out that we as a human are 99.9 made of the same DNA makeup. So the difference is only 0.1. And this 0.1 determine the sequencing and the order, and determine some diseases, some behaviors, even the color. As a human, we are consist of organs, and the organs are consist of cells, and inside the cells there are nucleus, and inside the nucleus there are nuclei, and then there are single central dogma. In the central dogma, there is what we call it the chromosomes, and these chromosomes are consist of more than 3 billion base pair. Now, if you stretch a single cell DNA, it will be around 2 meters long. And if you connect all the DNA together, it will, the DNA of your body will be around twice the diameter of the solar system. And you can imagine all these information are tied and organized in this small size. However, sometimes some mutations occur, and these mutations either to become insertion or deletion, duplication, translocation. And when a mutation occurs, then it will lead to a disease. As you can see in this diagram, where normal cells are divided, and for some reason, a mutation will happen. One of these types of mutation: insertion, deletion, duplication, translocation. The cells become abnormal, and that will lead to a disease. And as an example here, it's breast cancer. However, we have several thousands of diseases. As a geneticist, you have to know some few concepts. So I want you to learn today with me hereditary, trait, B degrees, genotype, phenotype, and homozygous, heterozygous, and we will go on these terms in details. But the importance of genetics came from the well-spread diseases all over the world based on genetic mutations or abnormality. So today, while I'm talking to you, we have over 6,000 known genetic disorder worldwide. And a study was conducted, and they found that 0.4 of the population have a single gene disorder. This is worldwide. And in terms of single gene disorder, we have 1 in every 50 people are affected with one of the single gene disorder. And 1 in every 263 people in the world are affected with one of chromosomal abnormality. So if this is the situation in the world, but what about our situation here in Saudi Arabia? In Saudi Arabia, and due to a high rate of consanguinity, we also have a very high rate of autosomal recessive disorder, and we have several diseases and mutations. And as an example, we have Down syndrome in every 1.8 per 1,000 birth. We have sickle cell disease prevalence range between 91 to 99 per 10,000, especially in the eastern region of Saudi Arabia. G6PD and other congenital heart diseases, and name it. And based on this, the Saudi Human Genome Program was initiated to solve this problem, because as you know that a patient with the genetic disease costs the country over $500,000 per year, and therefore it's a big burden in the country and in the health system, and therefore the country should do something, and therefore they established the Saudi Genome Program. And the aim of the program is to do the newborn screening test, to study some of the neurodegenerative diseases, loss of hearing, loss of vision, and so many diseases in Saudi Arabia. And the program was launched at KAUST, and as an outcome of this program, so far over 61,000 samples have been analyzed, and over 7,500 different pathogenic mutation. Also, we have over 25 different genetic panels and more than 134 publications, and 8 different centers. They are working together as one team. And now, after all these achievements, we can talk about the next generation sequencing. So the purpose of next generation sequencing as a new technology is not only to do some sequencing and discover diseases, but it has been applied in several categories and several things, starting from the infectious diseases. And as you all remember, when the COVID-19 hit the world, the pandemic time, the first thing scientists all over the world focus on to do the genetic sequencing for the virus. And when they discover the genetic sequencing, they start design vaccine. And the more mutations happen to that virus, the more people start develop several vaccines based on the genetic makeup of the virus. On the other hand, as I mentioned, over 6,000 unknown genetic disorder all over the world, including disability. So the people applying this technology, this new technology, to treat and to discover the root of cause for these diseases, and example of genetic disorder, Down syndrome, and other biomarkers for complex diseases such as cancer and cardiovascular diseases and other diseases. Not only that, but scientists start now thinking of drug discovery by using what they learned from the genetic sequencing and the genetic problems. With this in hand, now we move to the future actually by start thinking of the newborn screening test, start thinking of pre-implementation test, the BGTA, BGTM, prenatal diagnosis, and we are applying the knowledge of genetic sequencing in forensic testing and pharmacogenomics, pharmacogenetics, and as a tool for predictive testing in cancer and other complex diseases. In the world, there are 5 different categories actually to perform genetic sequencing. The first category is to use the single SNP sequencing. The second, and an example of this is the sickle cell disease. The second is the single gene sequencing. And an example for that is the cystic fibrosis, where we do the sequencing for CFTR gene, and a targeted gene panel, where we do the targeted panel to a target disease. And so far, we have offered 25 different panels here at King Faisal Specialist Hospital. And worldwide, there are several panels that can be used. And then number 4 is the whole exome sequencing. And finally, we can do also the whole genome sequencing. Now, based on this, we can target diseases, understand them, and do the analysis. Usually, when we do the analysis for sequencing, we also have to learn an important aspect, which is the pedigree. The pedigree is information about the patient's family, patient's history, and based on that, we can determine the mode of inheritance for that disease. And I don't want to go for complex things. I just want you to know that always square is a male and circle is a female. And if it's full, then that means it's affected. If it's half, then that means it's a carrier, and so on. And based on that, now we can go further step and understand how we do the sequencing. Some people, they think that if we want to do sequencing, the first step definitely is to collect a sample. So what the question now, what kind of sample? Some people, they think that we are limited to certain samples, for example, blood, saliva, fresh tissues, FFBE, and hair, filter card, and other. However, in fact, we can collect samples from any living part in the body and do the sequencing. Now, when we collect a sample, then every sample will have a corresponding kit for DNA extraction, which is the first step to do. For example, if you are collecting blood, then there is a blood kit, a kit for blood extraction, a kit for fresh tissue samples, for saliva, for FFBE, and so on. Once you get the sample, and before you start collecting the sample on DNA, you first have to get the IRB approval, sign the consent form, and start collecting the sample, and then do the DNA extraction. And basically, as simple as that, the blood sample will come to the lab in lavender tube. Then we will use the RPC lysis solution. Then we use the cell lysis solution, protein precipitation solution, and DNA hydrogen solution, and then RNase solution. And then, we follow the protocol and get the DNA extraction. When we get the DNA extraction, then we measure the DNA either by nanodrop or by the qubit. After that, we measure the quality for the DNA by blueprint, and then we prepare the library reconstruction, and we do emulsion PCR and enrichment, and then we do the sequencing. And this is the platform for the Ion Torrent platform. However, now the world is moving very fast. And if we have an Ion Torrent platform at King Faisal Specialist Hospital, nowadays there are S5, and the technology is moving very fast, where there is a machine called Genexus, which is the latest version of Ion Torrent platform. On the other hand, the next generation, Seek 550 from Illumina and other advanced machine. On the other hand, we have sequencing machines from MGI from China company. We also have other platforms from nanopore technology from Oxford and so on. But again, the technology is moving forward very fast, and more technology are expected to come, since this field become very important. Now, when we do the sequencing, we will have a huge data of variants or pathogenic variants, benign variants, and different variants. And therefore, we need a very special people, the bioinformaticians, who can do the analysis for this data. The system will give us a preliminary report, so we understand the quality of the data, and then we will receive a VCF file, or we convert it to X file, where we have several columns with a lot of information, starting from the name of the chromosome, from chromosome 1 to 22, then the X and Y chromosomes, then the point of the start of the mutation, the end point, and what happened, whether the A changed to T or C or G, and then the function, whether in the exome or intron or splicing, then the name of gene, then mutation, the effect, whether it's synonymous or non-synonymous, and so on. And usually when we do whole exome sequencing, we receive thousands and thousands and thousands of variants, and we have a criteria to filter the variants to get which variant is associated with the disease. And this filtration based on the quality assessment, variant as annotation, zygosity filtration, consanguinity filtering, variant functions, and mutation effects, and exosome aggregation consortium, and then we get the report. This diagram is to show you how we move forward with whole exome sequencing and finally get the right variant associated with the disease. Usually we get the whole family, parents, the affected index, and do the whole exome sequencing and start selecting the right variants. There are several international systems to report the variants. However, here at King Faisal, we are using the HGVS system, and this is just an example of how to report any identified variants. I don't want to go in details in this system. However, I put the link so anyone who's interested in this system can go and find more about the system. Now, since our talking is about the diseases and applications of genetic diseases, genetic sequencing, then I have to simplify things by mentioning that we have either a monogenic diseases, and monogenic is a disease that caused by a single gene, or we have a polygenic diseases, and these diseases, they have more than one gene that caused the problem, or genetic problem, or we have a chromosomal abnormality. And in this half an hour, I will focus on these three categories and talk about them in details. However, before I move forward, I want to grab your attention to that. The most killer diseases in the world nowadays, the people are looking for them from different angle, and they look at them as a genetic diseases, including cancer and including cardiovascular diseases. So the worldwide killer number one is the cardiovascular diseases, and now scientists and clinicians and people working in the clinical sectors, they start looking at this disease from the point of view of genetics. And you can see that more than 14 big diseases worldwide are listed as genetic diseases. Now, the mode of inheritance, we have several modes of inheritance. When we were in school, we used to learn that we have autosomal dominant, autosomal recessive, and so on. And I will mention that, and that is true. But today, I want to open your eyes that things are moving very fast, and therefore we have different mode of inheritance. And let me start with the autosomal dominant. In autosomal dominant, only one mutated copy of the gene will be necessary for a person to be affected by a disease or a genetic disease. And as you can see in my diagram, that 50% of your children will have chance to get that disease if you have an autosomal dominant disease. And there are lots of dominant diseases, and I just want to highlight example. It's a Huntington disease and Marfan syndrome, and some of the colorectal cancer diseases, and let us focus on only one disease, which is Huntington disease, autosomal dominant, and it's inherited and degenerative brain disorder, where the patient cannot control his movement, and therefore they call it a dance-like disease or a dance-like movement disease, because a mutation in a single gene, HD, will lead to uncontrolled movement of the patient. And here I want to grab your attention that, a single mutation in a one gene can change the life, not only for the patient, but for the family. And autosomal recessive. Autosomal recessive, two copies of the gene must be mutated for a person be affected by an autosomal recessive disorder. And it will be 25%, 25%, and you affected and unaffected, then 50% for the children to become carrier in this mode of inheritance disease. An example, sickle cell disease, and we have lots of sickle cell disease, unfortunately, in Saudi Arabia and internationally, and especially in African people. Then we have cystic fibrosis, we have other diseases, and in my presentation today, I will just have some few words about sickle cell anemia, since we have this disease actually here in Saudi Arabia. So in sickle cell disease, a mutation occurs only in very small portion of the gene in a known area, single gene, single SNP mutation, and that will cause this disease, and definitely it will change the life of the patient. The cells will become sickle in shape, and they go to the marrow veins and block the veins, and the patients will go on some episodes of collapse. And so this is due to a single mutation in a single gene. Another example is the cystic fibrosis. This disease is common in children, and a mutation in one gene, which is CFTR, and this mutation might take place at the beginning of the gene, or in the middle, or at the end. And as long as this mutation is pathogenic mutation, then it will lead to the cystic fibrosis disease. And believe me, I have seen several patients suffering from this disease and totally changed their life. Then we talk about X-linked, dominant and recessive. Now, as you know, as a human, we have 23 chromosomes. From 1 to 22, there are 1, 2, 3, 4, up to 22 pair chromosomes. And then we have the X chromosomes and Y chromosomes. Now, the X-linked chromosomes also cause the dominant one, caused by a mutation in a gene on the X chromosome. Again, we have several chance of the children to get that disease or to become a carrier or affected or non-affected. And Klinefelter syndrome is one of this disease. The disorder occurring due to a problem in X chromosome, and then that will change the life of male. And also we have an X-linked recessive diseases, and example for that is hemophilia A, Duchenne muscular dystrophy, and other diseases. And this is an example of X-linked recessive disease. The color blind, and this is something common more in male than in female. So 1 in every 10 males might get the disease, while 1 in every 100 males might have the disease. So please, I leave an example here. So if you can read all the numbers in the circles, then you are good. If you have a problem, then check. Now, again, I will go back to my introduction where I said we all learned that the mode of inheritance is always dominant and recessive. And now I will open your eyes to other concepts. As an example, we have a mode of inheritance called digenic inheritance. And that involves variants at 2 loci that are both required for the expression of the disease trait. The other mode of inheritance, dual molecular diagnosis, and involve variants at 2 loci that each is associated with the disease trait. Mutational burden, and here I want to grab your attention to what they call it expressivity and expression, severity. And I hope whoever want to dig in deeply in the genetics to go and read about expressivity and penetrance. Another mode of inheritance, incomplete penetrance. And sometimes you know that there are some mutations in the family and it appear in the first generation and then disappear in the second generation and then appear again in the third generation. And that is due to either expressivity or penetrance. Since we talked about the different types of mode of inheritance and genetic diseases, now we talk about chromosomal abnormality. And to simplify things, in chromosomal abnormalities, we have 2 types of chromosomal abnormalities. It's either numerical, so you have more than 23 or less, extra or less, or the structural abnormality where there is an abnormal structure of chromosome. So instead of the chromosome to become short arm, it will become long, or instead of become long, it becomes short, and so on. And an example of a chromosomal abnormality, it's a well-known actually disease, which is Down syndrome, a trisomy in 21 chromosome. And most of the people in genetic field, they understand and they can recognize this disease. Another disease, which is Turner syndrome, it's they also call it 45X or 45XO. And again, it's a genetic condition in which a female is partially or completely missing an X chromosome. Now, when we talk about this, again, technology is moving very fast, and we are trying to catch up, but things are moving very, very fast. all what I said, it's about the bulk sequencing. So what I mean by bulk, we get the blood or we get the tissue or we'll get with any samples and we do the, 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 the sequencing. What I'm going to say now is something totally new. 80% I'm sure of the audience never heard of. So it's very important to focus and listen about how the world now is moving. Now the world moved from bulk of sequencing, blood samples and do the sequencing and then do and get the result, to a more specific, more accurate, more precise sequencing, which they call it a single cell genomic sequencing. And now in the single cell genome sequencing, what they do when they collect a sample, for example, a blood, they do assortment by a machine, and then they segregate the different types of cells, and they take one single cell and they start doing a cloning of that cells a lot and do sequencing based on that single cell. And why this technology now is important and changing our life, not because they are using this technology for precise diagnosis in cancer and other diseases, but also in research, and it become used in developmental biology to understand some of the complex things such as the epigenetic role in diseases. Again, they use this technology in viral diseases and to segregate the heterogeneity of cells. Now, when we realize the importance of this technology, and although it's a new technology, we at King Faisal Specialist Hospital Research Center start thinking to accelerate scientific and medical breakthroughs by providing novel genomic information that helps in improving human health. Now, again, as I'm mentioning that technology is moving very fast. And here, what you can see is the 10x machine that we are planning to bring very soon at King Faisal. And that is the machine which will do the sort out of the cells and segregate the cells, and then it will give us a better understanding and insight for diseases and syndromes. And the purpose of using this technology, not only at King Faisal, but worldwide, is to foster special complex diseases such as cancer research, immunotherapy, and epigenomics, and metabolomics, and other hot topics in next generation. All the distance, and then based on that, they can determine whether the patients will have 21 chromosome, Down syndrome, 13 and 18, and other diseases. Nowadays, plus we have the fish, we also have the CGH, which is a little bit more precise, and people actually prefer to use this technique. And for the sake of time, I don't want to spend lots of time on this, but I want to highlight that there are different important differences between karyotyping and the fish. And also there are several important differences between karyotyping and the array. Now we can talk about sequencing in gene discovery, and we will talk about pre-implementation tests. For the pre-implementation tests, we have what we call it PGTM and PGTA. And basically, it's the pre-implementation testing for monogenic disorder. That is why they call it PGTM. And it goes through this 5 cycle, where we started with the consultation, then we move to the rest of the cycle until we get the result. This is different types of specimen where we can do the BOC samples, tissues, and we can do the amniotic fluid for the karyotyping. We can do the blood samples, and different sample will have different turnaround time. And this is due to, we have time for the sample to be cultured and so on. When we talk about genes and gene applications, then we have to think about how we can use the data coming from this sequencing. So scientists start all over the world thinking of gene therapy, how they can use this upcoming huge data and understanding for treating of diseases that couldn't be treated before. And from this concept, we have what we call it today, the gene therapy. And there are different types of gene therapy, including plasmid DNA, viral vector, bacterial vector, human gene editing technology, patient-derived cellular gene therapy products. And here I want to focus on what we call gene editing technology, as is one of the latest cutting-edge technology being used in gene therapy and basically, most of you maybe heard about CRISPR-Cas9, where there is an enzyme used as a scissor, come and cut the mutation, cut the DNA from the defected area or from the mutation or variant, and then the other DNA will be connected again and then repair that problem. Today, while we are talking here in Saudi Arabia, all over the world, there are several gene therapy drugs approved worldwide. Even though some of them are very expensive, but at least we can see the outcome and the applications of what we have done in the past and the outcome of the human genome program and projects. Now, with this, I will leave a question in your mind that, well, what is the future direction of the genetics? Since I highlight some words about gene therapy and I highlight some words about gene editing technology, CRISPR-Cas9. So where we are going to? We are going to have what we call it pharmacogenomics and pharmacogenetics. And basically, it's the right drug to the right patient at the right time with the right dose. And that will lead us to what they call it precision medicine. Precision medicine is instead of giving one drug to all patients with the same disease, instead of doing that, to design the drug or the therapy based on the genetic makeup for every single individual. So what we are aiming to achieve in the few upcoming years is to studying the genetic make up for every single person. And then when a person becomes ill with the disease, then based on her or his genetic makeup, will design the drug that suit that patient. And by doing that, we want to reduce the toxicity. We want to make sure that the increasing the efficiency of the drug and make sure that the drug is suitable for that specific patient. Where we are heading to. We are heading to an era where a new technology and nanotechnology is coming on. And we are heading to an era where intelligence genetics are coming. And we are heading technology where artificial intelligence is coming. Currently, while I'm talking, we have what we call it a robot surgeon at King Faisal here in Jeddah. People are doing surgery from America. So we are heading actually to a new era in medicine. We are heading for virtual reality, artificial intelligence, and all these things will move forward with your effort and hard work and the huge data coming from sequencing and single sequencing. And with this, I would like to say that molecular genetics, our latest wonder, has taught us to spill out the connectivity of the tree of life in such a palpable detail that we might say in plain words, this riddle of life has been solved. Thank you very much for your time, for your listening, for your attendance, and I will be more than happy to have any question you may have. Thank you. Any
Thank
question?
you for your great presentation. We received one question, if you can see from icon Q&A.
You mean in the chat? I couldn't see the question in the chat. Okay. No opening question. Q&A. So if you can, yes, thank you very much for excellent lecture. Yes, this is a very important question. Now, what I wanted to emphasize is that every population, they have different genetic makeup. We, in Arab countries, we have different genetic makeup, and we have different diseases caused by different genes. And therefore, instead of relying on American data or Russian data or any other international data, we start focusing on ourselves. And currently, and alhamdulillah, we have the knowledge, we have the machines, we have the technology, and therefore we start establishing ourselves. We have over 66,000 samples being processed inside Saudi Arabia for sequencing, and we, based on the Saudi population, we managed to have more than 25 different panels. So yeah, I agree with the person who asked that our population totally different than other population, and the Russian population is totally different than other American population, and definitely the mutations in Russian population or the causative mutation in Russian population definitely will be different than the causative mutation in America or in Europe or even other countries. Good evening, I have long. Well, I have a question here for someone who's asking about he or she is suffering from a problem, lung cysts, and it could be a tuberculosis, or it could be cancer, or so how the people can make the diagnosis. Usually, the diagnosis is started from the doctor, medical doctor. So the medical doctor will do several steps and collect samples, send it to the lab and do X-ray and collect different information from different sections, from different lab and on the screen. And after that, if he think that there is something genetic cause of this case, then he will send the sample to the genetic lab. In the genetic lab, if he is aware of the phenotype of the disease, then he will order a targeted panel or a specific gene, or in some cases, he will order a whole exome sequencing to understand what exactly happened. And before he make that decision, he usually go to the family and ask some questions about parents, grandparents, and grandfathers, grandmothers, and make sure that there is a history in the family where someone have the same condition. Yes, and again, I recommend the person who asked to start with the medical doctor, and the medical doctor will lead her or him to the right pathway. Yes. Any other question? Answer to yourself is there. For the recording, actually, I was facing some technical issues, so I'm not the right person to be asked if there is a record for this lecture or not. But I think the people who organized this wonderful webinar. I think they can ask and they can answer. And if they have if they recorded this session, then they can share it with you. Dr. Yusuf, do you recommend to do a panel of genetic testing? Yes. Now, I have a question from Ibrahim Saif that do I recommend to do a panel of genetic testing if in a family there are some, it depends on the type of cancer. For example, if it's a breast cancer in female or a prostate cancer in a male, and several people in the same age, when they reach to the same age, they get the same problem or the same disease, then yes, in this case, I suggest to go and do the right panel of genetic sequencing. And usually this kind of advice will be taken from the medical doctor. Any more questions? Omar, is the NGS best? Well, this is a very important question. And actually, the NGS is a cost testing, so it's not cheap, actually. It's very, very expensive. Even though when they start the Human Genome Project, they spend over 3 billion US dollar just to complete the Human Genome Project. And over 13 different countries participated in this project. Thanks God, thanks to the technology where the technology make these things cheaper. However, until today, we are talking about whole exam costing around 3,000 plus minus from one place to another, but still it's expensive for some people. And therefore, we cannot force people to do genetic testing unless they have to, unless they have some genetic condition in the family and within the family, and they have to do that test. They have no other options. Otherwise, it's not recommended for all the cases and all the patients. Yes. Any more questions? So there is a question here, how many types of NGS? Actually, all the sequencing technology in the world I have been exposed to in Saudi Arabia. So I would say that there is Illumina, there is Ion Torrent, there is MGI, there is platforms from China, from Germany, from America, and most of the technology actually exists now in Saudi Arabia. And from one place to another, they have different type of machines and technology. But Alhamdulillah, we have so many different types of machines and technology and platforms. So any more questions? I will be more than happy. Any more questions? So I think we left it only with 4 minutes. So if you have any more questions, I'll be more than happy to answer them. If no more questions, then I will leave the mic to Kunuz to conclude our webinar. And I hope you get something from my webinar. And again, please accept my apologize for the technical issues. And yes, see you again.